I want to thank all the speakers from this morning because they've really set up my talk. And luckily, I took out about 2/3 of my slides already, and then I should almost be on time. So we've heard a lot already about different modalities, some things about how they are useful to be talked about together. And I'm going to try to go over a kind of a overarching broad theme of how we can actually put these data sets together in a hopefully useful tool, and which we'll go through and do some live demos tomorrow morning to actually see how it really works in reality, not just in a presentation when everything's perfectly worked at least once. So I think this is another theme of our integrated workshop here, is really doing science over multiple scales with multiple modes of data, right? We have all these spectroscopic tools that give us these atomic resolution ideas of what's bound to what are the chemical species. We combine this with microscale to macroscale imaging to see how that varies over millimeters to, if you're crazy like I am, doing meters. And we really want to make some sort of large-scale global impact on something that started at a nanometer resolution, and now we're going up to the kilometer or continental resolution, right? So this is a huge challenge. How do we scale up this information from the nanoscale spectroscopy to the large-scale impacts? And integrating spectroscopy and imaging, I think, is a really key resource, and hopefully I'll convince you that this is all really fun stuff to be able to do. So I'm a lead scientist at SSRL, and I run our imaging program here. And this just shows you a flavor of some of our beamlines. You've seen some of this in Jocelyn's talk this morning. Large scale, small scale, low Z, everywhere. And so whenever I think of chemical imaging, I kind of come in from the X-ray centric point of view initially. And I'm glad John Barker came back in the room here, because this is probably the most over-measured grain of aquifer that's ever been done. And actually, John actually did some of these measurements. It's hard to believe that John was actually at the beamlines doing measurements. It is real. But we think about what Jocelyn was talking this morning, making measurements across an absorption edge to get chemical information from the spectroscopy point of view in an imaging sense. And effectively, we do this maybe 4 or 5, 6, 7 critical energies that can give us the contrast that we want for that chemical imaging. We can combine this with different methods. PCA might tell us what things are, where different chemical species may exist. The simplex volume maximization may tell us about end members, and we may do some linear combination fitting to give us the actual chemical species. And we can learn a lot about what the chemical environment is, in this case of iron, in this lovely aquifer grain, and where uranium sticks to it. And kind of one of the first examples of seeing uranium really sticking on to iron sulfides versus iron oxides in these kind of aquifer grains. And there's a really good test case to show how beneficial this kind of chemical imaging was. Now, I love X-rays, but there are other cool data sets too, other good modalities, right? These are often very complementary. As much as I want to think that X-rays can solve the world, there's lots of other things that are actually useful and helpful too. So we have physical microscopies, fluorescence microscopies. We can do fluorescent probes, histochemical markers if you're interested in biology, UV, infrared, Raman. We heard about electron microscopy. There's all these people here that do mass spec imaging. These are all really great complementary informations. How do we take all of these data and fuse them together, right? And learn more about our sample than the individual experiment, and kind of get greater than the sum of the parts. And that comes into our multimodal data collection. And so the challenges here are like, so, at SSRL, we've put some of these things together, right? So we put a spectrometer on the beamline. We have a nice camera that can collect really nice visible images too. And we can do diffraction on some of the beamlines as well. And if you do careful alignment, we can get multiple data sets that come out already pre-aligned, and everything's great. But it's really hard to put a mass spec on the XRF beamline, for instance. These are things that are never going to be done in line. We've thought about it, but it's just not really feasible. So now we need to think about how we're going to take different sets of data that are collected by mass spec, confocal microscopy, infrared, Raman. How can we actually put these together, right? And how do we register these different data sets to put them together? And this kind of started, one of the first examples of this came when I was working with some of my colleagues from Caltech, Woody Fisher. And he had produced this lovely data set that had some Archean pyrites, and he had some nanomagnetic work, or not really nano, it's micron scale, we'll say, SIMS, isotopes on sulfur. And we did some chemical imaging on the sulfur, and we found these really bizarre, weird sulfides. But it was one of these things where I had the samples, they were the same samples, but we're kind of like in PowerPoint and Photoshop, turning the data sets and trying to see where exactly was that point, how did it line up? And we eventually convinced ourselves we could find the same cross sections in there roughly, and that maybe things kind of agree, but it's really not the way you want to do things, right? And so this really began to think about how can we actually do multimodal work well, and combine things like isotopes with X-ray chemistry. And so we said, there have been years where we've all talked in various user groups about how we can make fiducial markers. And there's a lot of effort I think we have put into this at one point. And like, can we, some people come in with Sharpies. I mean, that's actually a really good fiducial marker when it comes down to it, because I can see it. I can see it in the X-rays. But it's, kind of crude, we'll say, right? We also had some people doing laser etching with the microscopes too, and they had the laser dissecting scopes. Those are great. And you can put down foils and gold things on your sample. But again, all these things, when you're doing multimodal data, you need to have a fiducial marker if you're going to use this that actually works in every mode of data. It doesn't. If I put down a Sharpie marker and I can't see it with the infrared, then it's not going to help me align that data set. Or so that's why we think about gold, but it just becomes a pain. So this is kind of the idea that we kind of started working with about a year ago or so. Whenever we're taking this imaging data, almost everyone takes a capture of the visible image at some point. They always want to line up, here's my pretty picture, here's my pretty data. So almost everyone has this visible image. And we can kind of make that the Rosetta Stone of our data regime. All we need to do is align those optical images, and those data cubes that are associated with that can be realigned, retransformed, and make a full data set. Now, it sounds great and easy, and it sounds like a great idea. It's a little more challenging in reality. I see some smiling faces there going, of course it is more harder than you think. But in concept, it's like, oh yeah, just take these pictures, match them together, and problems are solved. And it's just a transformation, right? It's just a mathematical transformation. There's lots of great software out there that does this. And you can even get the quality of your transformation by looking at how much things shear or have to rotate to make things work. And so, whenever you see things fail, you'll see a homography matrix failure kind of thing. That means they didn't align at all. But again, it is in concept, a simple idea of picking features align and then aligning your data sets. Once that happens, then we can fuse these together. So you've heard a little bit about the microanalysis toolkit, affectionately known as SMAC. And we've really worked recently to put all these formats together in a single platform. So again, the idea here is to remove this need of trying to overlay our data sets in Photoshop or PowerPoint, however you want to usually make your presentations look pretty. We take things from multiple synchrotron sources, both infrared, XRF, hard X-ray, tender X-ray, STIXM sources, multiple FTIR sources that are commercial, thermoadjuvant Bruker, multiple Ramons, multiple laser ablation formats, the lovely EMSL multi-language format that EMSL works with a lot, and even taking just random JPEGs and TIFFs that you might get out of your SEM. So all these things we can put together in the same platform and take all these different ideas and work with them together. So I'll give you kind of the state of where I am on this. So in reality, we're kind of in the multispectral world right now. That's what I would consider multispectral. I can load and process all these data sets. We can do some analysis on all of these data sets, and then we can kind of do combined, aligned, registered analysis on the results. And that's great. I mean, we can do things that we'll show you later on where we can compare how is the data from my infrared correspond to my XRF, and what variables that I've analyzed, how do they relate to each other. And that can be a really nice way of doing things that's not just again, overlaying images in PowerPoint. Where do I want to be with this? I want to be true hyperspectral, right? So we have a whole data cube of data that has spectral information, chemical information, UV, vis, Raman, mass spec, whatever it is, along all these axes, and analyze them as a full data cube of thousands of spectral points back, and do this full hyperspectral analysis. Now we need to do some slightly more advanced clustering techniques that can be used on this. We definitely need to work a little bit better on image and data registration. But we're not far off from this. It's been done before. So a lot of what I get to do is work with different communities. And so people in the cultural heritage community have done this with visible and infrared reflectance data, for instance. They did some clustering, did some really great analysis, but they did it in their own MATLAB scripts, which weren't shared with anyone else. And they did it once, and they wrote a paper on it, and they've never touched it again, right? And so it's like, well, why not? I mean, the paper was obviously really good, and just because it's not an easy to do process, right? And so we need better tools to really facilitate this. And if we have a better tool that's easy to use for the community will come and use those tools. So part of this is making these kinds of techniques accessible to our communities, not just, oh, look what I did in MATLAB once, right? And so, even Jocelyn referred to taking data across our multiple beamlines and putting them together. This has been something that we've always, oh yeah, I can just put the data together and it works great. But now we can actually do it natively in the program here. So here we have a potassium map at a large scale. There's straight bins. We can also fit them. Here's a part of a region of that map at the coarse resolution. And I can take a high resolution. Now this is now 5 microns rather than 25 micron data. And I can use the coordinates of both of those images and put them together. And now you have a little lovely inset of your high resolution map on your larger context. So we'll walk through this example tomorrow too. But imagine if this was chemical information data. You have a large context data file from one beamline, and you want to show your really cool high resolution chemical information. And we can put that in the same image so that your publications look pretty and nice, and not just trying to align it later on. And so I also talked about FTIR. Again, you might recognize this little fungal stem from Jocelyn's talk this morning. It's great to be able to load the data, but if you can't do some of the basic processing on the data, it doesn't really help you either. A good colleague of ours, Per Persson, has done a lot of really great work with FTIR processing in Python. Has a lovely program called Octaves, and it's really great for doing FTIR and Raman background subtraction and processing. So we can do all these things like water removal, has a really fast me-scattering correction, denoising and baseline. So we can take these total FTIR signals, look at the raw spectra, load those up, do some background subtraction, and extract that data, and then look at those characteristic peaks. And so you might recognize some of these figures similar to what Jocelyn was showing this morning. So we can do full analysis on this. So I took the full spectrum that we background subtracted, did a quick PCA on the full spectral characteristics, found 3 components that were interesting here. And it looks like you can denote the different FTIR signals in the stem and the hyphae around that. We can also integrate bands for cellulose, acid hydroxides, and lignin. And you see a very similar trend there. So our PCA is doing similar trends that you might get by actually integrating. And I can even cluster the entire data set on bins of 50 inverse centimeters. And you can see all these bits here with the proteins on the outside, the hyphae here in the different parts of the core, and probably a little bit of the double layer of tape somewhere stuck in the back corner even gets picked out by this, right? So using, again, crude clustering techniques, spectral analysis, not really always telling us exactly and identifying things, but telling us where we want to look for where things are different. And we can do the same process with our MALDI mass spec data. So this is, again, raw data that came out of the MALDI mass spec from one of the micro models, and shows you the silicon map on top and a potassium map. And then this is basically a factorization of the MALDI. I don't always like PCA because PCA gives you negative and positive things. That's hard to view. It's a pain. There's always one person that doesn't like PCA and they crash your paper for it. So this is non-negative matrix factorization, so everything's positive, so you can actually additively look at things. But you can even see like the 300 micron pores matches the 300 micron pores. So this is a direct overlay of those data sets put together. And then we can pull up these 3 different regions that the NMF pulled apart and see the different spectra in those locations. And even then, form together a movie then of going through M to Z. This is like from about 100 to 325. And you can kind of see how different compounds are showing in and out of here. I did a clustering on the same region, and you see these little areas around what we think are probably the bubbles in the micromodel, though, having different compounds in those. And if we look at a kind of a distance or a coarse distance from the center, you can see that some peaks are going up, some peaks are going down. So we're seeing a nice little trend around the different features in the micromodels. This is a Gaussian clustering. I hate k-means. This is a much cleaner, I think, clustering technique. But again, it's a really crude way to begin to get some rough ideas of what's happening on it. And we do the same kind of thing with the FTIR data. We did the same kind of thing. We can do the same kind of thing with our XRF data. We can use those same exact methods with all of these techniques to look at them individually and see how things are changing. So I mentioned the auto registration. So that great optical image. This was one of my very first attempts at this. And I took this is actually a coral sample from Colleen Hansel. She left it behind at SSRL. And so after she's left it around for a month or 2, it becomes mine. And I was like, oh, I need to do something. Let me test it out. And so we got the total infrared signal, the total calcium signal. It's a coral. That's obvious. It should be, right? And, I love my XRF visible image. I don't love the way that my FTIR microscope tiles the data, so I can actually see the edges. I don't like that, but that's the vendor stuff. And it turns out that the resolutions were vastly different. So if I put those 2 images together, one's about this big and one's about that big. And I try to relate them together, and it finds points. Resolutions aren't matched well at all. And that's what my warped image looks like now. And you might guess that really doesn't look anything like this at all, right? And so that was definitely a failure of the transformation. Definitely not a fine transformation. 3,000% shear. That's really not going to work. So you try a couple different things. I can blow up the resolutions. It still doesn't work, right? And so I did fall back and say, okay, I can draw a bunch of dots on one image. I can draw a bunch of dots on a second image. Don't have as many dots as the machine vision tries to analyze with, but as long as I put at least about 10 or 12 dots in there, I can do a pretty good job. And it did it pretty well. Got a good overlay and a good matching. One of the next things you'll see I won't promise it in 3.10, but 3.11 for sure, we'll have better ways of drawing and segmenting things to actually make these alignments better. But then, this is actually an interesting challenge too. This shows that stem again. And Jocelyn even hinted at the fact that there were some slightly different methods in between the infrared and the visible, but you can still see the visible data looks good. That's also the visible data from the other beamline. You can see that things have changed even between beamline 7 and beamline 14, and definitely from the infrared. But we can put this all into the defining all the points around the edge of the stem and put it all together. And it still works pretty well. You definitely have better hyphae in the infrared, but we get the stem to match pretty well. Now, there is a little bit of a change of the angle over here, so it doesn't do the best overlay on the far left side. But even for something that's kind of been tweaked a little bit, we actually get a pretty good overlay. And then we can do multimodal analysis with this. So now this is now taking the X-ray data from phosphorus and sulfur in this particular instance, along with all the infrared peaks that have been assigned to things like cellulose, protein, lignum, acid, hydroxides, and amines. And we can do a factor analysis. This is basically it's like PCA, but it's doing PCA on the covariance rather than the total variance. So it tells you what things are actually alike, not where things are different in that sense. And you can look at those factors and we can see how it identifies different parts of the hyphae in the stem there. And this actually shows you that the XRF data trends together along with the lignins a little bit is very similar because it's only in the center. And the cellulose and the protein lignins are different from both of those. So again, yeah, I would have liked to see that the X-ray data was a lot more aligned with some of the infrared stuff, but it is a really nice way of showing you how these things, three groups are separated and different from each other. And we can do this and we'll go back to that coral. I know I'm jumping between samples, but I'm kind of cherry picking the best examples of what works best for this particular example. One of my favorite things to do in chemical imaging and now this total multivariate analysis is this end member characterization. Because that's often what we care about. Not only just what's there, but what's the most different from something else, and how can I characterize those end members then to characterize everything in the sample. So this is a geometric end member usage. It's faster than non-negative matrix factorization, and it actually comes out of the remote sensing literature. Simplex volume, we'll call it maximization today. I can't remember if it's maximization or minimization, but it doesn't matter. In any case, again, it looks for real data sets, real data points that characterize the most different things. Now, the real problem now is we have, say, a data set that has visible data from the camera, infrared data from one microscope, and our XRF data. We've integrated that together and aligned them, but the scaling is drastically different. My infrared signal has really nothing to do with my XRF counts I get on my detector. They're different detectors, different processes, completely different. So when we do our multivariate analysis between multimodals, we are actually able to independently weight all n number of data sets. So I can cluster all the visible data and weight that evenly with all the infrared data and weight that evenly with all the XRF data. Or if I want to bias it, I can see if I bias one more than the other, how that affects my clustering analysis. This is, again, really important because the different scales can really drastically determine how you do a multimodal analysis. So when you do this, though, we see that carbonates correlate with the calcium in the XRF. That's great. But there's actually two factors in the infrared that kind of go with the carbonates. And when I look at iron contribution, there's actually three different infrared signals that correlate with iron, showing you a iron carbonate in the center and two different types of organic matter relationships on the outside of the coral. So I can do all this with just infrared and XRF. I don't even need iron XAS to tell me that I have different iron species. And we can also do the same thing now with clustering. We kind of showed clustering with the mass spec data, clustering with the infrared data. And you can kind of see as we bring in more modes in this Gaussian clustering, I get one cluster that's only XRF based. If I add in some of the visible data from the cameras, I see some other features involved, and I bring it all together, I see organic layers from the infrared becoming distinguishable from the blank spaces or the iron areas. I see the different cracks present. And so this is now a full cluster analysis with all three modes of data together. And you can do fun things and overlay them visually too. And this actually then shows you another challenge because you see that the infrared data is coarser than the visual data, and my XRF data is somewhere in between. Some of the blockiness of those pixels comes out from that particular cyan color of a cluster is really dominated by infrared information versus some of the other ones on the interior, which are dominated by the visible or XRF data. Now you can look at that in the component factors too, but you can kind of see it right away when you look at the clustering. Actually not bad on time. So where do we go from here? So this was all bin data that I then processed together. And again, that's a multispectral kind of a way. We're coming really close to now being able to analyze these as a full data cube, as a hyperspectral analysis. And right now we're binning resolution, so extrapolating with cubic by cubic sampling to change your resolutions. It's not bad, but we want to think about better how we can intrinsically treat these different resolutions in these data sets together. Again, smarter analysis for data mining. There's a lot of good stuff out there, different types of clustering that work way better than the old-fashioned PCA analysis. And luckily, I mean, I'm pretty good at computing, but I don't need to invent the wheels again. There's lots of great tools kits for people to use. And it's not that you need to learn how to use them. We just need to make them accessible and easy to use, right? And that's what we're doing with this here. Again, kind of going back to what Jocelyn showed, we have a really good question of like, how do we time these measurements? What's the order of measurements? Is one going to damage the sample? Is one of the preparations going to damage the sample? So we need to think about a workflow of what we do, when, and how to avoid artifacts. So that when we do these registration techniques, we're not like losing. I have no more hair to lose, right? So I can't pull my hair out anymore. So how do we avoid these problems, right? And then finally, what modality do you like best? If you have one that you'd like to see, let me know. I spend a lot of time making data formats work. It's a lot of fun, almost fun. But if you have data that you'd like to see in this kind of work, let me know. We'll make it work. And then lots of people to thank. Lots of great ideas have come from lots of our collaborators. Lots of people here provided data. You saw a lot of it earlier on. I really need to thank an incredible SSRL imaging program that I have. Sharon, Nick, Jocelyn, and Eleanor, all being awesome support people. Manny's a new member of this group. Welcome him to the group too. We also have great staff that support all of our stuff. Dave, Doug, and Bart. And there's a whole bunch of people here that pay some of my bills here and there. They're all very important. But these are all the things that we get to do, all the people that get to help us do what we want to do.